I'm part of Objective 5, the biotic group, and our objective is to look at the distribution of biotic factors like pathogens, insects, earthworms, weeds, and really look at how those relate to present climatic factors and then to try to project those into the future. So I'm going to just show you an example of how we're trying to work with this on a data set that a grad student derived a few years ago. And that was to look at a disease called Fusarium crown rot and then to relate that to 30-year averages. So a little bit of background on this disease. This is called Fusarium crown rot. It's a fungus that is in the soil, will attack the roots, and then will move up the stem and cut off the flow of nutrients to the top. So it ends up significantly reducing not only the yield, but also the quality and the test weight of the crop. So here you can see a picture of the bottoms of the stems being rotted away. So this is very widely distributed across the wheat dryland areas. A couple of other things that are important to remember about this disease. It's caused by a complex of 2 different fungi, so it makes it even more complex to look at. The reason we chose this is because this disease is really influenced by drought stress and excess of nitrogen. If you have a drought stress, the fungus will move right up the crown and will cause very severe disease. And then finally, this pathogen survives on crop residues, and so that's another component, and it can also live in the soil for a very long period of time. So basically what we did was to sample over a 2-year period 100 locations throughout all the agro-ecozones. From each location we did 5 samples, so that's a total of 500 samples each year. What we did was to look at the disease rating at each of those locations, and both crown rot rating and node rating. So we look at how far up the pathogen moves. And then we also plated out specimens to see which fungus was going to colonize those. And then for the kind of modeling thing, we use generalized linear mix models to try and look at the relationship between these variables. And the one that I'm going to show you just as an example today is something called factor analysis. And factor analysis is very similar to principal component analysis in the sense that it's a variable reducing, where you try to take all these independent variables that you have and reduce them down to just a few that can be used to predict. And the other neat thing about this method is that unlike principal component analysis, you can use it to define trends. So this just shows you the 2 factors that we derived, and these are some of the components that went into it. And this first factor is heavily loaded by temperature variables. So mean temperature of the last 30 years, mean temperature in coldest month, mean temperature in warmest month. And then we have a second factor that's heavily loaded by the precipitation variables, in this case highly loaded by snow and or by rain, and to a lesser extent by snow. So now that we've derived these factors, let's see how our disease relates to these. And so what we're showing here is factor 1. So this is that factor that's heavily dependent on temperature. So the further we move on the x-axis, the higher temperature each one of those locations are. And then we're looking here at the dependent variable, which is the level of disease. And you can see in this year we had a very good relationship where the higher temperature sites we had higher levels of disease. And this kind of fits our hypothesis that we had. Then we look at the second variable, and we found that this one really was not as predictive. In fact, the first year, 2008, there really was no significant relationship between the level of disease and the rainfall, and that's indicated by this line here. But in the second year, 2009, which was a higher rainfall year, we did pick up a significant relationship. So this indicates to us that really we need to not only be looking at 30-year averages, but also the year-to-year data, because that's so highly variable in trying to predict this disease. And then finally, one more example. This is looking at the distribution of the 2 species and how they respond to factor 1. So again, this is the temperature factor. And we found that really these 2 fungi have very, very different ecological niches. And this is something that we had suspected based on previous studies. So here we see this is the probability of isolating Fusarium pseudograminearum. So as you go to higher temperature locations, higher temperature wheat fallow, low rainfall, you get a much higher ability to isolate that. But on the other hand, Fusarium calmorum kind of seems to have an indirect inverse relationship. So we find it more at the lower temperature sites, higher elevation sites. So that was also an interesting thing that we were able to pick up with this type of factor analysis. So just in conclusion, we showed that those 2 species have very different niches. We think that the pseudograminearum is in warmer temperatures, lower elevations, whereas the calmorum seems to be prevalent at higher elevations with greater moisture. And this is an approach that we can use to really look at how these diseases are distributed across broad geographical regions, and then use that to try to predict future climate change models. So we're going to be doing the same type of approach with other data sets. We have a big nematode data set that we're working on, and then 2 or 3 other species that we're going to be doing a very similar thing. So again, our take-home lesson here is, can we figure out how these pathogens are distributed? How do they relate to present-day climatic factors, and can we model those in the future to see what's going to happen? So that's all I've got.